Mhm. Full screen is var. Okay, I'm going to switch to English now. So thank you for attending this lecture. I'm honored to be here and presenting this topic. I'm Luka Nadiradze. I'm a neuroscience master's from Ilia State University. I also have a bachelor's degree in computer engineering. I'm a software engineer right now at one of the research institutes based in Netherlands. And over there, I've been working on several research and development projects involving neuroscience, not only neuroscience, sound engineering, and so on. So I'm going to talk about not my experience, but like what I have learned about EEG and what you can expect from EEG recordings and EEG experiments. So hopefully after this lecture, you will have a general idea what it is used for, what it is good for, and different terminologies and concepts that you need to learn if you want to work in this field. So the field that I work in is generally called neuroscience, but there are many adjacent topics like psychology, cognitive psychology, and so on. And I'm more of a like technical guy who works on EEG signal filtering and so on, but I still need to understand how it's produced and the biology of it and so on. So I'm gonna start just by discussing different ideas that the general population may have on EEG and whether or not they are true or not true. So first of all, just to be clear, I'm talking about this device that we all probably have seen or known or heard about. And on the right side here, this is the typical EEG recording. Okay, so this is like brain waves. We read brain waves from our scalp, and then we can infer some things from it. So this is a usual neuroscience experiment and how it looks. So I'm going to start first by asking questions. I think you can answer because I don't need you. I don't need to unmute you. You can, I think, talk by yourselves. So these are interactive questions. I know they are maybe simple or like very, very surface level questions, but we need to like ground ourselves first when we're working with EEG. So first question would be, if I put this EEG device on your head, could I figure out what you're thinking about at the moment? So if anyone would like to answer, please go ahead.
Can I?
Yeah, sure.
No, no one can figure out what I'm thinking, but they can figure out how can I respond to some stimulus, like when someone doing a Stroop test and like clicking something on keyboard. That's the recording
Yeah,
they get from EEG, but they couldn't figure out what I'm thinking or what other person is thinking.
exactly. That's true. And basically this like general questions, what you're thinking about and so on, can't be answered by EEG, but generally what you have described also is like you can design experiment cleverly, then to EEG help you infer what you're paying attention to. And this basically can give you like a good amount of information, but you need to have an experimental setup, and it's not like a real-time decoding of your thoughts. So what about this one? Like, can you infer which color you were thinking about? Like, let's have a narrower domain, or which number you're thinking about.
No, I don't think so.
Yeah, so if I say I'm thinking of a general number, can you figure out which number is it from EG? The answer is no. Like you can't say like it's 33 or I'm thinking of color blue or something. But again, if you design an experiment, and we'll get to this in such a way, then you can actually do this. But not this strongly, not like this strong, thing. So next question is more, I think, simpler and easier to answer. Whether your eyes are open or closed, what do you think? Can EG answer that?
Yes, EG can answer that. I observed in the lab doing EG, and they put like, I don't know what's called in English, I don't remember, these devices around the forehead, and they can figure out if your eyes are open or closed.
Oh, okay. So maybe you're talking about ECOG, not ECOG, sorry, EOG devices, like actual um, actual electrodes that measure if your eyes are
Yeah,
closed or not.
electrodes. Yes, yes,
Yeah.
yes.
That also works, but basically they're very similar to EG. They are basically EG,
Yeah.
but they're used for that purpose. So you can actually, not only from that electrodes, generally observing brain activity, you can infer it from over here, back of the head, from over there too. And can you tell whether you're asleep or not?
Yes.
Yes. And what about like what you're dreaming about? I
Uh,
think
I don't,
it's harder than figuring out what you're thinking about.
uh, I
To
don't
answer it
know.
more.
I don't think like the topic of what you're dreaming about, but if you're anxious during sleep or like having a nightmare, like they can figure out
Yeah,
that.
yeah.
By
What about
brain waves,
actually?
I think.
Yeah, what about sleep phases? We know we have different sleep phases like light sleep, deep sleep, REM sleep. Can we differentiate those using EEG?
Uh, as far as I remember, yes.
Yeah, that and EEG is basically one of the most used devices for like sleep research. And yeah, answer is yes. And whether you saw something surprising or not, I'm going to answer this as yes, probably, because EEG is most used also in attention experiments when we are measuring attention. And whether I saw something surprising, this means I'm directing my attention to that. And probably the answer is yes. Your deepest secret, yes or no? Probably.
No.
No, but whether you're lying or not, like lie detector.
No.
Yeah, I also think no, when you are asking like this, but there are some experiments that you can design that can help you deduce if you're lying or not. And maybe we will have time to talk about that as well. And whether you're concentrated on a task, this I can answer that this is true. You can measure it by looking at the brain waves. And also some people think like EEG electrodes put electricity into the brain, but that's not true. We are just reading the electrical activity from the brain. It's a totally passive device. It's not putting anything into the brain, but there are also some devices like transcranial magnetic stimulation device that can put actually electrical stimulation in the brain and that can induce electrical activity in the brain. But EEG is like totally, first of all, safe and non-invasive, and it just reads the activity of the brain that already is happening. So then I'm going to talk about the like basic characteristics of an EEG signal and how it's produced and how we read it and which parts of the brain are involved. But it's going to be like very simple in very simple terms. So first of all, what we need to know is that EEG device measures the potential difference on our scalp, like the voltage on our scalp that originates from deep inside our brain and also from cortical activity, like more surface activity of our brain. So this is measured in tens of microvolts, which is 1 microvolt is like a millionth of a volt. So it's very small signal and we need very precise instruments to detect that. And this signal is mostly, as I said, generated from the cortical activity, like the surface of the brain that has wrinkles. And this is generated not by a single neuron, like we cannot measure activity of a single neuron on the scalp. It is generated by like billions of neurons activating together, like populations of neurons. And what's more important, the synchronous activity of those neurons, and that we can detect. But it also dissipates from the source. Like when it's generated, then it has to go through the bone and scalp and skin and everything. And so this signal dissipates. So what we are measuring is just a fraction of that signal. And of course, we cannot also record the activity from deep inside the brain, like thalamus, for example. Those are like basic things that we need to know. Like it is from cortical activity, it is from rhythmic patterns arising in the brain, and it's from multiple billions of neurons. And these EEG signals, because they are so small, they can have like artifacts from other electrical activities going on in the body, for example, like muscle movements, blinking of the eyes, and so on. So that is one of the biggest challenges when you're working with the EEG signals. So EEG is one of the ways to measure the brain activity. We also have fNIRS, fMRI, PET scans, different brain scans, but EEG is good in one thing, which is temporal resolution, meaning you can measure activity in the brain in very small time frames, like sub-second and sometimes sub-millisecond ranges. So like brain activity is very fast and things change in the brain very quickly. So one of the most reliable ways we found to do that is using EEG. So it has high temporal resolution. It's one of the strengths. It's non-invasive because we are not surgically getting into the brain and doing any kind of operations. It's very cheap. We can have like commercial EEG devices like those we see here, which are not available, like counterparts of brain imaging are not available at this cheap. Like fMRI costs a lot, MEG also costs a lot, and so on. And it's also portable, like at least some of them. But the downsides are, as I said, low spatial resolution. Well, I actually didn't say that, but low spatial resolution means we are not sure where the signal comes from. So we are sure when the signal is, but not about where. So where in the brain, it has low resolution. fMRI and other methods are more precise as to where the signal comes from in the brain. Okay, so there are multiple ways to record EEG. So there are wet and dry measurements. So these are like different configurations. I'm not going to go into the details, but different configurations of how you can record EEG. And also there are multiple ways to put the electrodes on the scalp, but we have standardized everything. In most of the experiments, you need a specific way to put electrodes on your head because everybody has different head shape. So we need to like come up with some kind of standard to make experiments comparable. One of the like parameters that EEG devices have is sampling rate, which is important. Maybe also it's like number of channels. That's also important. And I'm not going to talk about anything else for now. So number of channels is basically just how many electrodes we have. So how many places from the brain are we reading the signal? That's the number of channels. And sampling rate is how often we reading the signal like voltage from the brain. So this means like 250 hertz means we are reading voltage from a single electrode 250 times in a second. So the higher this sampling rate is, like naturally it's better, and higher the channels, we have more information. But it's also how we do the experiments and so on. So guys, if you have any questions, also you can ask me during the lecture. So do not hold back if you are interested in any certain topic that I'm going over. Okay. So what is this? This is like 1020 system, which is like a top-down view of this one is a top-down view of our head, our scalp, simplified. And these different numbers we're seeing is different positions on our head that we can put electrodes onto. And all of those have like meanings. For example, this one means frontoparietal, C means here central, this means parietal, this means occipital. So different regions on the brain, and there are specific relationships of these electrode placements that we need to follow in order to make a good EEG experiment. So for example, this nation means nation and Indian is places on our heads where we have like a nation is this place between eyes, between forehead and the nose, that we measure all of the electrodes from, distance of the electrodes. And Indian is another extremity, like the bone of the skull, like in the back of the head, like there is a small dent here, and that's called Indian. So this is basically what you will need to know, and how to do this measurements and so on. You should also be able to like read EEG signals and just know what is what. So here, as you see, these are channel names. So placement locations are channel names also. And here you see a typical EEG recordings, and you can see the same channel names here, like arranged vertically. And horizontally is just EEG recording that is recorded over time. And yeah, what do we see here? Like we see, okay, let me ask you, like, do you think that all of this is coming from the brain, all of this data?
Some of them might be noise.
Yes. Which ones do you think? Do you think this dent, like deflection, is from the brain or from some other noise
Maybe
source?
muscle, like blinking or something.
Yeah, it is actually from blinking. It is blinking artifacts. And when we look at the place where it is, it's prefrontal, so it's most close to the eyes, and it's logical for it to be from the eyes. Like when you see the channel name, I don't know if you can see it or not on my video, on my screen, but yeah, these are prefrontal electrodes. And also, if you look closely on the bottom here, it's not EEG, it's like ECG, like cardiogram, and this is heartbeats. And it's often we do like measure other biomarkers along with EEG, like heart rate, respiratory rate, eye movements. What else? I don't know, like anything involving experiments. During the sleep, we measure those different movements as well. Also like leg movements. And that's so. What are alpha waves? Have you had like experience with it? Do you know what it is? Have you heard it? Okay, so alpha wave, like we obviously know for different brainwaves, right? And these different brainwaves have different names. And one of them is alpha waves, which is like one of the first types of waves that we observed and like discovered. Somebody was saying something. You can say it. Like it's also written here, so I don't know. It's like alpha waves have this different like characteristic visual, like photos like visual compared to the over there on the right. So these ones are usually when you close your eyes, you can observe these alpha waves in your brain. So this means you have your eyes closed, you are relaxed, but you are still conscious. You are alert. You're not sleeping. This one means your eyes are open and those are called like beta waves, which means you are just engaged. You are doing something. You are doing some task, or you are mind wandering or something. Like you can infer general states of mind just by looking at EEGs, but other than that, you need some more tools. Okay, what about sleep? Okay, so first of all, we can measure sleep states. Like basically the definition of sleep states, I think, also comes from the EEG. Because what is light sleep, and when does a light sleep become deep sleep, and when does deep sleep become REM sleep? You need to define this, and basically EEG helps us define those concepts because EEG is different in each of the sleep stages. Do you guys know usually how many sleep cycles we go through the night? Or how long it's 4, and how long one lasts usually, like one full cycle?
I
2 hours.
I think it depends. Some less, like half hour, and some more.
Like
I remember
I
exactly.
remember like full cycle is around 90 minutes, if I'm not mistaken. And
Okay.
if you multiply that by 4, it's like roughly average of how much we sleep during the night. But it changes during the night. So like first REM sleep, first REM stage is shorter and it gets longer and so on. But yeah, I'm just gonna say that EEG is used in sleep research a lot, and it's a useful tool. We can see the theta waves when we are first going into the light sleep. Then when we are getting near the deep sleep, we see the slower waves. We also start to see sleep spindles and K complexes that are specific shapes, which are associated with like memory consolidation and so on. And then we have slow wave sleep, also called deep sleep. And this is how it looks. It's delta waves. It is very low frequency waves and high amplitude also. And this is like restorative sleep stage, associated restorative sleep stage. And you can actually not tell REM sleep from the waking, if I'm not mistaken. Only way you can tell it is like you're not moving any muscles. So you're completely paralyzed. You can move only your eyes, but all the rest of the muscles are paralyzed. And that's how you can tell being in a REM sleep versus being fully awake. Okay, do you guys think we have free will? Like we are, yeah, this is like very difficult question, I think, and it's also like goes into philosophy and so on. But there are some group of scientists that actually argue that you can read from the EEG some actions that you are about to perform, even before you know you are going to perform it. So they designed this experiment. You may know this guy Vsauce from YouTube, and he has this TV show from like 9 years ago. And in one of the episodes, they did this experiment where you are asked to press a button, and machine learns your EEG patterns when you're pressing the button, and after some time, it lights up the button before you press it. So even before you know that you are going to press the button, the system knows before you, and you can't press it before it's lighting up. So you can't lie to it basically, because it allegedly reads subconscious waves before you actually are aware that you decided to do some task. Does it make sense?
Yes, it does.
Okay, so there are many discussions about implications of this and whether or not this experiment is legit or not and so on, but it's certainly an interesting experiment, and I encourage you guys to look at it if you are interested. And also from the same episodes, we had a lie detector basically from the EEG device. So lie detector, before I said, can you detect lies or not? Generally, lie detectors are not reliable devices at all. And they obviously can't be used in court. But you can recreationally use them or whatever. So one of the episodes had this experiment where different participants stole different items. No, actually, one of the participants stole a watch and others didn't. And then they went out to find out which participant stole that item. So basically, one of the participants who stole the watch and knew what the watch looked like had different EEG pattern than the other participants. So in this case, Hannah was who stole the watch, and she didn't think of anything when she was shown the different jewelries. But when the watch came up, on this watch specifically, she had a specific reaction, which we could tell because we also had control jewelries, and this they measured this amplitude for this watch, and it was basically larger than other participants, and that's how they found out that she was the thief, quote unquote. And we actually repeated this experiment, me and my friends, and this actually worked with high confidence. So you can do this, but for this you need to have a constrained environment, set number of items. You can have multiple, thousands of items, and you have to ask a question more concretely. Does this has this person seen this item before or not, and so on. But yeah, any questions about those two experiments? Okay. So next, I'm going to talk about brain waves in more detail and what do we usually use for working with brain waves, and what's next? Yeah, and different kinds of experiments we can do with EEG. Okay. So if no questions so far, I'm going to continue with this concept maybe that we all heard, and I'll be also talking about this during this presentation. For example, deep sleep is associated with delta waves. Meditation also has some associated waves, theta waves are associated with meditation or altered states of mind. Alpha waves are eyes closed awake, beta waves are eyes open thinking, and gamma waves are more concentrated thinking on some really hard task. But how do we name the different waves, and why do we name those waves, and how can we actually analyze those waves from the EEG? That's the question. So when we record EEG, we don't record that in frequencies. We record that over time. So we measure voltage over time, and this is what we see here. So these are different channels, and we measure channel activity over time. But this time activity for a single channel is consisting of multiple different frequencies. And these different frequencies, like different sine waves, basically, and these different sine waves have different amplitudes. And when you add them up, you can approximate any kind of signal, any kind of EEG signal or any other biological signal or whatsoever. So you can look at these time signals the other way, like which frequencies are in this signal. Does it make sense? Like when you add up, like sum all the waves, then you get this original wave, but you can like decompose any signal into different frequencies. There's a whole method and whole subject. It's called signal processing. You're going to learn it in the university also if you're like in computer engineering or software engineering or whatever, but it's actually really a cool concept. It comes up in many different subjects. We use, just to say it, and I'm not going to go into details, we use Fourier transform to decompose a single wave into multiple sine waves, multiple frequencies. And then we can measure which frequencies are most present in a wave. And if, for example, if alpha frequencies are present more, this means like we are relaxed and so on. So it is just another way of looking at the data, and it's sometimes more useful, especially in doing neuroscience. So when you see the graphs of where we have frequency on x-axis instead of time, then we know it's a frequency domain. And this comes up in many different experiments, or like if you're reading EEG papers, they're doing that. Or if you guys know about like brain-computer interfaces, or if you're interested in that, you have to know these things. And there's
Sorry
also
for interruption. I think we have less than 1 minute in this link, so I think it will end and then we can re-enter in the same link.
Oh, okay.
Okay.